Hey people, so this lesson I'm going to take you through cellular respiration at the VCE level, and what we're going to look at along the way is the inputs and outputs of cellular respiration, the process of cellular respiration and where each input and output is used or produced. We're also going to have a chat about the mitochondria and look at its structure. And then we're also going to have a chat about anaerobic cellular respiration and some factors that affect the rate of cellular respiration. To start us off, like photosynthesis, you want to make sure you're familiar with the equation for cellular respiration. And like I said with photosynthesis, if you know the equation, get it down on your piece of paper, on your SAC or your exam as soon as you can, because it can help give you some hints or help dig you out of a hole when you get to a question where you can't remember the inputs or the outputs. It's not a bad little prompt. So I haven't got the equation written up here. That is your task to do that. So if you were to pause it now and try and write it, and then let's imagine you've now come back, and what you would hopefully have is your inputs on the left hand side of your arrow. Your inputs, also known as your reactants, would be 6 oxygen plus glucose would produce 6 carbon dioxide plus 6 water plus ATP. And the amount of ATP would be dependent on the type of cellular respiration taking place. More often than not, we talk about aerobic cellular respiration, and it makes 36 to 38 ATP. Beautiful process, the old cellular respiration. Why do we need it? We need it to make ATP. So ATP, adenosine triphosphate, we visited back in photosynthesis. It is the only form of energy that a cell can directly use to carry out all the processes it needs to carry out to keep you alive. At the end of the day, if you did not have ATP in your body, you would be dead. So every living organism undergoes cellular respiration because every living organism needs to make ATP. They don't all undergo it in the same manner, but they all undergo respiration nonetheless. So it's a process that makes ATP, and to make ATP, we break down foods that we eat, and although we can make it through technically breaking down carbohydrates, fats, or proteins, in VCE Biology we just focus on how we break down carbohydrates in the form of glucose, and by oxidizing glucose into carbon dioxide and water, that releases energy, and then that energy is used to resynthesize ATP from ADP and inorganic phosphate. The mitochondria is definitely where the action's at when it comes to cellular respiration. So you would no doubt refer to the mitochondria at some stage in your life as the powerhouse of the cell. Though that is a catchy little phrase, it's not going to do you much good at this level of biology. So we would prefer to say mitochondria is the site of cellular respiration, which is the site of ATP production. Now, it's not the case that all cellular respiration takes place in the mitochondria, and it's not the case that the whole process of cellular respiration takes place in the mitochondria either. But for aerobic cellular respiration, most of that process does take place inside the mitochondria. For anaerobic respiration, it doesn't take place in the mitochondria at all. And that could have something to do with the fact also that you have bacteria that would undergo anaerobic cellular respiration. And since they are the size of a mitochondria, well, they can't really have a mitochondria inside them. So let's have a better look at this organelle. So your mitochondria, it has its outer membrane, and then it has this highly folded inner membrane, and the folds are referred to as cristae, and what the folds are doing is they're increasing the surface area within the mitochondria. When we have a look at the process We'll talk about 3 stages. We'll talk about glycolysis, which occurs in the cytosol. We'll talk about the Krebs cycle, which occurs in the mitochondrial matrix, which is the internal space in the mitochondria. And then we'll talk about the electron transport chain, and it takes place in this inner membrane. So by having all of these folds and increasing that surface area, it's more room for more electron transport chains, so it increases the amount of ATP we can make. So main structures we're looking at are outer membrane, inner membrane, intermembrane space, which is the space between the outer and the inner membrane. That'll be important when we get to the electron transport chain and do our best to confuse the hell out of you about hydrogen ions moving in and out of the intermembrane space. And then, like I said before, the inside of the mitochondria is the matrix. Like chloroplasts, we apply the endosymbiosis theory to mitochondria, and as a way of explaining how they may have ended up as organelles inside cells. And that endosymbiosis theory says that at some stage in their evolution, a large eukaryotic single cell would have engulfed a mitochondria, which at that stage would have been a type of bacterial cell. And then once engulfed, it became more specialized inside that cell along with other organelles. So some of the pieces of evidence we use that suggest this mitochondria could have at one stage been a bacterium is the fact that it's got DNA, because not all organelles have DNA, only the mitochondria and the chloroplasts. Its DNA is naked DNA, so it's not enclosed in a nucleus, which is also the case for bacterial cells. The DNA is circular, which once again is something it has in common with bacterial cells. The size of the mitochondria itself is also similar in size to the average size of a bacterium, and the mitochondria also has ribosomes inside it, which once again is similar to a bacterium. So it's a pretty self-sufficient organelle. It has DNA, so it can make messenger RNA. It has ribosomes, so it can translate messenger RNA into proteins, and the genes and proteins that are in the mitochondria are pretty much exclusively dedicated to making the enzymes needed for cellular respiration to take place. So we've got 2 main categories of cellular respiration, both of which make ATP. Aerobic cellular respiration, which means in the presence of oxygen, and anaerobic respiration, which means making ATP in the absence of oxygen. So whether it's aerobic or anaerobic, we're still breaking down glucose. It's just that aerobic respiration has more stages but produces a higher yield of ATP, being 36 or 38 per molecule of glucose, whereas anaerobic respiration is a simpler and quicker process, but it only has a yield of 2 ATP per 1 molecule of glucose. So we'll have a look at both of them. So let's look at aerobic cellular respiration. We'll break this process down into 3 stages. Stage 1 is referred to as glycolysis, and this stage takes place in the cytosol, so that's the part that does not occur in the mitochondria. Stage 2, once glycolysis is done, we have the Krebs cycle, and that takes place in the mitochondrial matrix, so that internal part of the mitochondria. And then stage 3, the electron transport chain, which, as I said before, takes place in the inner mitochondrial membrane. So I'll do my best to talk you through it in the following slides, but it would also be important to note that also on the same YouTube channel, or you can get the link through Compass, is a animation that's breaking down and drawing the process of cellular respiration in more detail. So that would definitely be worth a watch as well. So here we go. cellular respiration, aerobic cellular respiration. Stage 1, glycolysis. So this occurs, though this says cytoplasm, you're better off saying cytosol would be more accurate. Maybe try and write it a bit neater than that. Takes place in the cytosol. So in glycolysis we have glucose coming in, and you can see here glucose is a 6-carbon sugar. It's a sugar made up of 1, 2, 3, 4, 5, 6 carbons. Glycolysis is the process where glucose is broken down into pyruvate, and pyruvate are 3-carbon sugars, and because we started with glucose, which was a 6, we produce 2 molecules of pyruvate. So to break glucose down into pyruvate, it would be a series of chemical reactions taking place. You don't need to know those chemical reactions, you just need to know that glucose is broken down into 2-pyruvate during glycolysis. Now in doing that, we're actually able to produce some ATP, and we can produce 2 ATP in glycolysis. And also, by breaking glucose down into pyruvate, we release some hydrogen ions and some electrons. And we have our coenzyme here, or our carrier molecule, NAD, which is the cousin to NADP+, which we used in photosynthesis. They have the same role though. So NAD comes in and it picks up the hydrogen ions to become NADH. So in total, glycolysis breaks glucose down into 2-pyruvate. That enables the production of 2 molecules of ATP, and it also enables the production of NADH as an output. The ATP will get used by the cell. The NADH will travel to the electron transport chain. Here's a little consolidation task for you. So have a go now at putting in the inputs and outputs of glycolysis, and you can also draw yourself a little diagram of the process over here. So hopefully what you've been able to identify as the inputs of glycolysis are glucose, and the corresponding output to glucose as an input would be our 2-pyruvate. So input is glucose, output 2-pyruvate. Another input would be ADP plus PI, and an output would be ATP. And our final input would be NAD, our coenzyme, our carrier molecule, and its output would be NADH, which is it in its loaded form, having picked up the hydrogen ions. So look, you could as an output list H plus as a hydrogen ion, and you could list electrons, but it is much easier and more streamlined just to show the NADH, because by writing that, that incorporates the fact that you've produced the hydrogen and electrons, and that tells whoever's reading your answer that you understand that's what's happened. Stage 2 of cellular respiration. So once glycolysis is done, the pyruvate moves into the mitochondrial matrix, and then it undergoes the Krebs cycle. Now, the Krebs cycle, as you can see by our picture, we have this thing called the link reaction first, and then under the link reaction we have the Krebs cycle. So as per VCAR, you could include everything shown in this diagram. You could sort of explain that as the Krebs cycle. So for the Krebs cycle, you could say pyruvate enters the Krebs cycle, is broken down into carbon dioxide. By breaking it down into carbon dioxide, we produce 2 ATP. We also release hydrogen ions and electrons to produce NADH and FADH2. And that would be it, done, all coming under the Krebs cycle. However, in reality, it is a little bit more complex than that, and the pyruvate does not enter the Krebs cycle directly. So now I'll talk you through the process that includes the link reaction. So stage 2 of cellular respiration, like I said, pyruvate has entered the mitochondrial matrix. The pyruvate is then incorporated into a coenzyme called coenzyme A. So we've taken our pyruvate, and one pyruvate at a time is combined with coenzyme A, and in doing that, one carbon dioxide molecule is produced and some NADH is also produced. What we're left with is a molecule called acetyl coenzyme A, acetyl-CoA, which is essentially the coenzyme which now has the 2 carbons attached to it because one was already lost. Acetyl coenzyme A delivers those 2 carbons into the Krebs cycle, and then those carbons are further broken down into another 2 molecules of carbon dioxide, and by doing that we're able to produce ATP. We're able to release hydrogen ions and electrons to produce FADH2 and NADH. So if you follow one single pyruvate, we're producing 1 CO2 in the link reaction, and we're producing 2 CO2s in the Krebs cycle. But of course we've got 2 pyruvates, so if we're producing 3 carbon dioxides per pyruvate, well, we're obviously producing 6 carbon dioxides for 2 pyruvates. So Krebs cycle gives us a net yield of 2 ATP. Consolidation task for the Krebs cycle now. So you know the drill. Draw yourself a little diagram. And I should have mentioned in that last slide too that the FADH2 is a cousin to NAD and to NADP+. And along with NADH, FADH2 delivers its hydrogen ions and electrons to the electron transport chain. So Krebs cycle, and we'll include the link reaction in this as well. In terms of inputs, our inputs would be 2 pyruvate. Another input would be ADP plus PI. Another input would be NAD, and another input would be FAD. In terms of our outputs would be 6 carbon dioxide, output would be 2 ATP, output would be NADH, and another output would be FADH2. Notice that I told you how many carbon dioxides were produced and how many ATP were produced, but I didn't tell you how many NADH or FADH2 were produced. And the reason is you don't need to track those numbers. You definitely need to track how much ATP is produced in each stage, and you definitely should refer to the quantity when we're talking about the inputs and outputs from the equation, but you definitely don't need to keep track of how much NADH and FADH2 we're producing. This here is a closer look at the Krebs cycle, and you definitely 100% do not need to know this diagram. It's just here to give you a little bit more detail at what that process is. So essentially what the Krebs cycle is a series of chemical reactions that takes the carbons delivered into it by acetyl-CoA and converts them into a number of different forms, essentially just breaking it down into carbon dioxides. Stage 3 of cellular respiration. So we've had glycolysis, then we've had the Krebs cycle, and now we've got the electron transport chain. And it is definitely the more confusing stage to explain. At the end of the day, you don't need to know it in heaps of detail, but it's one of those things that's a bit hard to explain in less detail, but we'll give it a crack. So electron transport chain takes place in the inner membranes of the mitochondria. So you could also say in the cristae of the mitochondria, or those little folds of the inner membrane. And electron transport chain is the stage that produces most of our ATP during aerobic cellular respiration. So we produce 2 ATP in glycolysis, we produce 2 in the Krebs cycle, and in the electron transport chain we can produce 32 to 34 ATP. So it's almost like the other 2 stages are there just to set up for the electron transport chain to work. And here's what happens. So think of it as a chain. So that's what this picture here is trying to show you. Think of it as a chain that is in the membrane. And what these little cogs really should represent are little proteins that are being used to transport electrons down the electron transport chain. So to the top of the chain, our carrier molecules, NADH and FADH2, which have come across from glycolysis in the Krebs cycle, they come to the top of the electron transport chain and they drop off their passengers, being hydrogen ions and electrons. The hydrogen ions would be sitting just on the inside of the inner membrane in the matrix of the mitochondria. The electrons would be delivered into the electron transport chain, which is in the inner membrane. So the electrons fall down the chain to the bottom of the chain. And they fall to the bottom of the chain because sitting at the bottom of the chain is oxygen. And oxygen is a very electronegative molecule, which means it has a strong affinity for electrons, and the electrons are drawn down the chain to combine with the oxygen, and that's what happens. So the electrons fall down the chain and combine at the bottom of the chain with oxygen. Now, sitting in that matrix is our hydrogen ions, and what we do is we use the energy produced from the fall of the electrons to pump the hydrogen ions from the mitochondrial matrix across the inner membrane into the intermembrane space. That's all good. So the reason we do that is because also sitting at the bottom of the chain is a molecule called ATP synthase, which is a protein, and it's an enzyme and a transport protein all in one. And what ATP synthase does is it catalyzes the chemical reaction that converts ADP plus PI into ATP. However, we need to make ATP synthase work. And the way it works is it's activated when it has hydrogen ions diffusing through it. So let's go back a step. We have pumped the hydrogen ions from the mitochondrial matrix into the intermembrane space. And we've kept pumping them into the intermembrane space so that we've got a concentration gradient, whereas the hydrogen ion concentration in the intermembrane space is a lot higher than the hydrogen ion concentration in the mitochondrial matrix. Now, natural laws of diffusion will mean that the hydrogen ions will want to diffuse from high concentration in the intermembrane space across the membrane to low concentration in the mitochondrial matrix. However, the only way the hydrogen ions can pass through the inner membrane is by traveling through ATP synthase. So because we've got the concentration gradient, the hydrogens move through ATP synthase back into the matrix. By moving through ATP synthase is activated and it can catalyze the ADP plus PI into ATP, and we're producing our ATP. Perfect. Now, what we're left with is we have a need to maintain that concentration gradient, because as soon as the concentration gradient stops, the hydrogen won't go through ATP synthase and it won't work. So as the hydrogen ions travel through ATP synthase and enter back into the mitochondrial matrix, they actually combine with the oxygen that was sitting at the bottom of the chain to form water. And what that does is, we say it mops up the hydrogen, it removes them from the mitochondrial matrix so that it ensures the hydrogen ions can never accumulate in the mitochondrial matrix. So we always have the concentration gradient where there's a higher concentration in the intermembrane space so that they keep moving through. Like I said, it's pretty involved, and it's hard to follow with me just talking it. Jump onto that other YouTube clip I made and have a look at me drawing through the process. And obviously your teacher will go through it in class as well. In simple terms, if we dumb it right down to something that you could write for a 1 mark or 2 mark question, we would say during the electron transport chain, hydrogen combines with oxygen to produce water and maintain a hydrogen ion concentration gradient. This enables the activation of ATP synthase, which is used to produce 32 to 34 molecules of ATP. And that'd probably get you out of trouble. Consolidation time. So inputs and outputs of the electron transport chain. Your inputs of the electron transport chain, the things coming into it, are oxygen, NADH, FADH2, and ADP plus PI. So they're all of your inputs. Your outputs, the things that are produced during the electron transport chain, are water, NAD, FAD, because they're the unloaded acceptor molecules since they dropped off their hydrogen ions and electrons. And your final output would be ATP, and it would be 32 to 34 ATP. So that was aerobic cellular respiration. Let's now have a look at anaerobic respiration. So anaerobic respiration is the cellular respiration that takes place without using oxygen as an input. And we have 2 different types depending on the organism it's taking place in. So we have alcohol fermentation, and you commonly would see that taking place in things like yeast, which is a fungi. And we also have lactic acid fermentation, which takes place in different organisms, and it actually takes place in humans as well. So essentially, anaerobic respiration is just a modified glycolysis. So that should tell you, where does it take place? In the cytosol. Doesn't use the mitochondria in any capacity and doesn't use oxygen in any capacity. So in comparison to aerobic respiration, what they have in common is that they both use glucose. They also have in common the fact that they both have glycolysis as a stage. They both have in common that they produce ATP, but a difference is anaerobic respiration is only ever going to produce 2 molecules of ATP per molecule of glucose, whereas aerobic respiration will produce 36 to 38 molecules of ATP per molecule of glucose. In terms of their byproducts, anaerobic respiration will produce carbon dioxide as a byproduct. It can produce lactic acid as a byproduct, and it can produce ethanol, which is alcohol, as a byproduct. Woohoo! Aerobic respiration, your byproducts are carbon dioxide and water. So lactic acid fermentation. So this is an anaerobic respiration that takes place in some fungi, some bacteria, and it can also take place in us, in our muscles. So if you're doing like a 400-meter race or an 800-meter race where you're going max intensity exercise, and then your legs feel that burning sensation at the end of the race, that's because you've got lactic acid building up. Why would the human body undergo cellular respiration without oxygen when we can also do it with oxygen? Well, if we're exercising at an intensity where we're using ATP at a faster rate in which we can make it by using oxygen, then we've got to make it somehow. And we can make ATP a lot more quickly if we're doing it anaerobically. Problem is we can't make as much of it, and we can't make it for as long, and we produce byproducts that affect our body as well. So lactic acid fermentation, let's have a look at the process. So we start with glucose, and because it's a modified glycolysis, the glucose is broken down into 2-pyruvate, just like normal glycolysis. And by breaking glucose down into 2-pyruvate, we produce 2-ATP and we produce 2-NADH. Now here's where it changes. Normally your NADH would travel to the electron transport chain where it would be unloaded, and then it would be able to return as NAD back to glycolysis to pick up hydrogen ions once again. Now, for anaerobic respiration to work, it's important that the NADH is able to be unloaded somewhere. So what happens, because we don't have oxygen, the electron transport chain won't work, so it won't unload it. So we go in lactic acid fermentation, we go glucose, break it down into pyruvate. By doing that, we produce the 2-ATP and the 2-NADH. The pyruvate then accepts the hydrogen ions and electrons from NADH, so it unloads it. And by accepting that, the pyruvate actually gets converted into lactate, which is our lactic acid, and that's why we produce that as a byproduct. The benefit of doing that is our NADH has been unloaded, so we now have NAD again to then accept hydrogen ions and electrons from breaking down another glucose molecule. So it can keep going and going. You probably only be able to do that for about 60 seconds though, because in that time the lactate will have built up so much that it starts making your blood too acidic, and that affects your enzymes working. Alcohol fermentation is really similar to lactic acid fermentation. So once again, it's a modified form of glycolysis. And alcohol fermentation takes place in yeast, which is a type of fungi, and there's also some kinds of bacteria that undergo alcohol fermentation as well. Here's how it works. So we start with our glucose once again. Glucose is broken down into 2-pyruvate once again. In doing that, we produce the 2-ATP and we produce the 2-NADH. Once again, pyruvate unloads the hydrogen ions and electrons from NADH. But whereas last time it became lactate, this time that molecule releases carbon dioxide. So there is one of our byproducts. And what is left is a molecule that we call ethanol, which is our other byproduct. So in doing that, you would have a net yield of 2 ATP per glucose molecule, and your outputs, your byproducts, are ethanol and carbon dioxide. Because cellular respiration is a process that's governed by the availability of its inputs, and also takes place due to the enzymes in your body, there's certain things that will affect the rate in which cellular respiration will take place. So the temperature will affect the rate of cellular respiration, the availability of glucose, so the more the better, and the concentration of oxygen. So for aerobic respiration, once again, the more oxygen, the faster the rate of cellular respiration will be. If we graph the effects of temperature on cellular respiration, what we see is the exact same trend we saw when we looked at the effect of temperature on photosynthesis, and the exact same trend we would see when we look at the effects of temperature on enzyme activity. So the rate of cellular respiration will increase as temperature increases up until a point, and that point is where it's reached its optimum temperature. Cellular respiration will be taking place at the highest rate. Past that point, cellular respiration would drop off very suddenly and get down to zero, because cellular respiration relies on enzymes, and enzymes have their optimal temperature, and we all know that past that temperature the enzymes denature, and if the enzymes denature, cellular respiration won't take place. In terms of glucose availability, you would have a linear relationship between the concentration of glucose and the rate of cellular respiration. So as the concentration of glucose increases, the rate of cellular respiration would also increase. It would get to a point though, like our photosynthesis graphs, where you would get a plateau, and the point where it plateaus, what we would say is another limiting factor that's not the availability of glucose, is restricting the capacity of cellular respiration to take place. So at that point, it doesn't matter how much glucose you add, cellular respiration won't take place any faster, and maybe that could be because the availability of oxygen is being the limiting factor there. I suppose glucose, it's sort of like a substrate, so think of it as you're going to get to a point where all of the enzymes in cellular respiration are already taken up by glucose. So even if you keep adding more and more substrate, the rate of reaction won't increase anymore. When we look at oxygen concentration, it's pretty much the same as glucose concentration really in terms of its trend. So once again, linear relationship exists. Increased oxygen concentration will be an increased rate of aerobic cellular respiration up until the point where we get our plateau. At that plateau, another limiting factor is restricting the rate of cellular respiration that's not oxygen. Could be carbon dioxide, for example. So any further increases in oxygen past that point of plateau still won't increase the rate of cellular respiration. This little box down the bottom here says, note, in facultative anaerobes, the rate of anaerobic cellular respiration will increase with a decrease in oxygen concentration. So we've got some organisms that undergo anaerobic respiration. That can be facultative anaerobes, which means they can do anaerobic or aerobic respiration, a bit like us. And then we have what we call obligate anaerobes, and they're organisms that can only undergo anaerobic cellular respiration. In our facultative ones, the ones that can do aerobic and anaerobic, well, their rate of anaerobic respiration would increase with a decrease in oxygen concentration, and their rate of anaerobic respiration would therefore decrease with an increase in oxygen concentration, because the body's typically going to favour producing ATP aerobically when it's got the choice.